Hello friends, in the previous video we were talking about immunology, and we have talked about the innate and acquired immunity. What is the cellular difference between innate and acquired immunity? What are the types of acquired or adaptive immunity? We have also talked about the maturation of T lymphocytes in the thymus. We have talked about the positive selection, negative selection, and then we talked about that both the T lymphocyte, after getting mature from thymus, they will get enter into the lymph node, the secondary lymphoid organ, and the B cells, B lymphocyte, after getting mature from the bone marrow, they will go and reside in the germinal center of lymph node, secondary lymphoid organ, and they will stay there until they are being activated by the APC. When the APC will reach there, the APC will convert uh, T lymphocyte and B lymphocyte. APC will make them active. It will cause the proliferation and differentiation of lymphocytes. So how the T lymphocytes and B lymphocyte will get proliferated, we will talk about in today's lecture. This lecture will focus on the proliferation and differentiation of T lymphocyte. In this lecture, we will talk about how the Th0 will be converted into Th1 and Th2. In this video, we will also talk about the cell mediated immunity and humoral immunity. Okay, so stay with me. So for example, this is a lymph node. Okay, in the lymph node, in the center of the lymph node, T helper cell, also called as CD4 plus cells, or cytotoxic T cells, also called as CD8 plus cells, they will come after getting mature from the thymus, and they will reside in the central part of the lymph node, while in the periphery there are some specific structures that are called as germinal centers. Germinal center. In the germinal center, our B lymphocytes, after formation from bone marrow and after getting mature in the bone marrow, they will come and they will stay there. Both the T cells and B cells, they are not active yet. They are not active. They need to be activated, and they will be activated with the help of antigen presenting cell. What is antigen presenting cell? As I have explained in the previous class, that whenever there is an infection in any tissue of the body, for example, this is a tissue, and there is an infection in any tissue of the body, in every tissue of the body there are some specific innate cells are present, like macrophages, neutrophil, dendritic cells. They are always present there. Okay, and some of these cells are called as APCs, antigen presenting cells, and the example of APCs are, I explained, macrophages, dendritic cells, and B cells. Now these APCs are responsible to engulf the invading foreign particle. They will engulf it. They will cause the phagocytosis of the engulfed pathogen, and then the small fragments, for example, this is a macrophage, it will engulf the foreign particle like bacteria, and it will digest it, dissolve it, and when it will dissolve it, it will present the fragment of this bacteria on its MHC-2, protein. MHC-2 protein, major histocompatibility, is a protein that is present on the outer surface of the APCs, and it will present the antigen on its surface with the help of MHC-2. And now this APC, with the help of lymph vessel, it will come into the secondary lymphoid organ, and in the secondary lymphoid organ, this APC will cause the activation of T cells. It will cause the activation of T cells, T lymphocytes. Okay, and then it will also then some of the T lymphocyte will also cause the activation of B lymphocyte. But in today's lecture, we will focus on the activation of T lymphocyte. We will discuss the activation of B lymphocyte in the next lecture. Okay. So, by the way, this is a very short summary I explained right now. The detail of this antigen presentation on MHC-2 has been explained in the previous lectures. Okay, so if you want to know more about this process, how these APCs are going to present this fragment on the MHC-2, you can visit my previous lectures. Okay, well, as I have explained, that in today's lecture we are only going to focus on the activation of T lymphocyte, so I am going to remove these germinal centers, because we know that in germinal center basically B lymphocytes are present. Okay, now germinal center have been gone. Now let's talk about the Now let's start the discussion of activation of T lymphocyte. As I have explained, there are basically two types of T lymphocyte, uh, CD4 plus cells or T helper cells. They are called as CD4 because on their outer surface there is a protein is present that is called as CD4 protein, cluster differentiation 4 protein. So these are the reasons they are called as CD4 plus protein. The cells having CD4 proteins are called as CD4 plus protein. Other than CD4 plus protein, they will have some other protein that is called as T cell receptor. Okay. Similarly, now let's talk about the cytotoxic T cells, and these cells are basically CD8 plus cells. If they are CD8 plus cells, so it means on the outer surface they will be having a protein called as CD8 protein, and other than CD8 protein they will be having another protein called as T cell receptor. So T cell receptor will be common in both of the cells, but the difference will be that a T helper cell will be having CD8 to CD4 protein, while the cytotoxic T cell will be having CD8 protein. Okay, they are mature but they are not active. They need to be activated with the help of APC. Okay, now let's see. Let me tell you one more thing. That this CD8 protein, it can get attached with major histocompatibility type 1, and CD4 protein can get attached with MHC-2. Okay, why I am telling this I will explain in the later part of the video. Well, what will happen? So for example, this is APC, okay, and on its outer surface it will be having MHC-2. On the MHC-2, we know that the fragment of antigen will be present. Okay, they are called as APC. It means they are going to present this antigen to the T cells. So what will happen? The T lymphocyte Th0, CD4 cells or Th0, with the help of T cell receptor, they will get attached with the MHC-2. They will recognize this antigen and they will get attached with the MHC-2. So this is stage 1 reaction. Stage 1 reaction in which MHC-2, CD4 protein and T cell receptor get attached, and this reaction is further strengthened with the help of CD4. CD4. We know that CD4 protein is present on the T helper. So, and we know that CD4 can get attached with the MHC-2. So this is MHC-2, this is CD4, so they can make bond, they can get attached. So this is the first stage interaction in which the major proteins are being formed. Major proteins are being formed. Now, there is another protein that is present on the APC, and this protein is called as B7 protein. Okay, B7 protein. B7 protein will express on these APCs when they will engulf and phagocytose the foreign pathogen. Okay, so when they will engulf the foreign pathogen, the expression of B7 protein will be started, and it will express on the APC. Now this B7 protein will be recognized by another protein that is present on the helper T cell or T cells that are present in the lymph node, and the name of this protein is CD28. CD28. B7 protein is basically a complex of two proteins. The one is CD80 and the second is CD86. So now these are the co-receptors. These are the co-receptors. So the stage 1 reaction in which major proteins were involved, and in the stage 2 reaction the co-proteins are involved and they are getting attached with each other. So this is the stage 2 reaction. But there is another type of reaction, third stage, third reaction. Now what will happen in the stage 3, that when these core protein and these major proteins will get attached, there will be another protein that is called as CD3 that is present over there, and it will be attached with both CD4 and with the T cell receptor. This protein is called as CD3 protein. CD3 protein is basically responsible for signal transduction. What will happen? This, for example, I am removing this T lymphocyte. I am saying that this is the nucleus. So what will happen with the help of this signal that CD3 will send to the nucleus, the gene present in the nucleus will start expressing more prominently two genes. The one gene is specific for the interleukin 2, and other gene is specific for the interleukin 2 receptors. Okay, both these interleukin 2 and interleukin 2 receptors are basically protein. So obviously they must be having some gene on the DNA. So both of the gene will start expressing after CD3 will send signal to the nucleus, to the DNA. So when this there will be expression of interleukin 2, so it means there will be formation. First, obviously, if there is expression of interleukin 2 gene, it means now after the transcription, messenger RNA will be formed, and then messenger RNA will come out of the nucleus. It will get attached on the ribosome, and protein will be formed, and that protein will be interleukin 2. So interleukin 2 will be start forming, and interleukin 2 will start releasing from the this TH0. Okay. Now at the same time, there is another gene that is coding interleukin 2 receptor will also be expressed by the signal that is being sent from the CD3. So interleukin 2 receptor proteins will also be formed, and these receptors they will present on the surface of the T cells. Now what will happen? This interleukin 2 will come and it will act as an autokine. Autokine, any substance that act on the same cell from which it has been released. So interleukin 2 has been released from this cell, and at the same time it is acting on this cell. So it means this interleukin 2 will be called as autokine. Okay. So now this is stage 3 reaction. Stage 1 reaction in which major proteins were involved. stage 2 reaction in which some co-receptors were involved, and stage 3 reaction in which cytokines like interleukin 2 were involved. After these three type of reaction, this T cell will get activated and it will start proliferating. It will start dividing. 1 to 2, 2 to 4. It will start dividing. Now what will happen? We know that when it will start proliferating, majorly it will get converted into two type of cells, Th1 cell, and it will start converting into Th2 cell. Okay, now point is how this Th0 will get to know that I have to convert into Th1 or I have to convert into Th2. This is very important point. How will this T0, this now I am removing the nucleus and I am writing here Th0. This Th0 will get converted into Th1 and Th2. Well, let me tell you briefly the function of these two, then I will explain how this Th0 will get to know that either I have to come into Th1 or either I have to convert into Th2. Okay, the function of Th1 is that it will activate the cytotoxic T cells. And cytotoxic T cells, these cytotoxic T cells, it will go and it will activate the cytotoxic T cells. And the cytotoxic T cells, you know, they are responsible for the intracellular pathogens. It will kill the intracellular pathogen, the pathogen that have been entered into the cells. Now antibodies will not be able to neutralize those pathogen effectively because they have entered into the cell. So to counter the intracellular pathogen, these cytotoxic T cell will help out. Okay, so it means Th1 will activate cytotoxic T cell, and Th2 it will activate B cells that were present in the germinal center, as I explained earlier in the lecture. They will activate B cell, and B cell will make antibodies. And antibodies, so it means antibodies are basically responsible for the extracellular pathogens. So it means if the pathogen is extracellular, this Th2 will work. And if the pathogen is intracellular, we do not need to produce antibody, but we need to activate our cytotoxic T cells so that it can go and it can act. How this will act on the intracellular pathogen and how antibodies will neutralize and opsonize, all these things, detailed mechanism will be discussed in the later video. In today's lecture, actually basically I am talking about the activation of T cells, so I am not going to the depth mechanism of these cells. Okay, so what I mean to say, now if we need to activate cytotoxic T cell, it means Th0 should convert into Th1, and if we want to if we want to counter the extracellular pathogen, Th2 will have to convert into B cells so that antibodies can be produced. Okay, but again my point is how this Th0 will get to know that the pathogen is extracellular and intracellular. Either I should activate Th1 or either I should activate Th2. This is a very important point. So now let me tell you one thing. This thing will be explained by this APC. This is very intelligent cell. Very intelligent, very clever. For example, if this APC engulf or phagocytose intracellular pathogen, it will release some different cytokines. And with the help of those cytokines, this Th0 will convert into Th1. But if this APC engulf extracellular pathogen, so it will release some different cytokines, and it will convert Th0 into Th2 so that antibodies can be formed. Clear? Okay, now let's see. For example, if the pathogen is intracellular, this APC will release interleukin 12, and there will be some receptors for interleukin 12, and when this interleukin 12 will act on the nearby Th0, this Th0 will start proliferating into Th1. Okay. Now this interleukin 12 is called as autocrine. Now this is called as paracrine. Autocrine is when the substance released from one cell is acting on its own receptor is called as autocrine. But when a substance that is released from one cell and act on the nearby cell, those substances are called as paracrine. So this is autocrine, and this is the paracrine. So what will happen when interleukin 2 will release from this substance, will act on these receptor, so it will guide Th0 that you have to convert it to Th1, because the pathogen I have engulfed, I have phagocytosed, is basically intracellular pathogen. So we need to activate this pathway. We need to activate cell mediated immunity. Okay, you know that the immunity that is being mediated by these cytotoxic T cells is called as cell mediated immunity, and the immunity that is being mediated by antibodies is called as humoral immunity. Okay, so this APC will tell this Th0 that the pathogen I have engulfed is basically intracellular pathogen. So that is why I am releasing interleukin 2 to tell you that I have engulfed intracellular pathogen, so that you may activate cell mediated immunity, you may activate cytotoxic T cells. Okay, similarly, if it has engulfed in extracellular pathogen, it will release some other cytokines. In most of the book it is written like we don't exactly know which cytokine will activate this is responsible to convert Th0 into Th2. So they will write that X cytokine, because research is still going on. But in some book it will be written like interleukin 4. Interleukin 4 will be released from APC. It will act on its receptors. And it will guide this Th0 so that it may convert into Th2, because the pathogen is extracellular, so we need to activate humoral immunity. We do not need to activate cytotoxic T cell mediated immunity. Okay, now so Th1 has been activated, Th2 has been activated. So Th1, let's first talk about Th1. Th1 will release three major substances. It will release interleukin 2, it will release interferon gamma, and it will release tumor necrosis factor alpha. Okay, now Th2 has basically two functions. The one I have explained in the previous part of the lecture, that it will activate this cytotoxic T cell, CD8 cell. It will activate the cytotoxic T cell. This is the first function of Th with the help of this interleukin 2. Now, why this? It is releasing tumor necrosis factor alpha and interferon gamma. Okay, this Th1 will basically, after activating cytotoxic T cell, it will migrate toward the tissue, infected tissue, in which pathogen has attacked. Okay, so for example, this is the infected tissue. Okay, and assume that this is the nearby blood vessel capillary. Okay, so this is the infected tissue and this is the nearby blood vessel. So what I am going to talk about, that Th1 will reach into the infected tissue. So with the help of, here it will release interferon gamma and tumor necrosis factor alpha. With the help of these cytokines, it will enhance the activity of macrophages. It will enhance the activity of macrophages, point number 1, and it will transmigrate Transmigrate meaning the WBC, the white blood cells, macrophages, monocytes, neutrophils that are present in the nearby blood vessel. You know that all these cells are present in the blood. So it will attract these cells. How these cells will be attracted? Detailed mechanism will be explained in the later videos. Okay? How what is the role of integrin protein, selective protein, and ICAM, PCAM, all things will be explained in a detailed manner. So, but today's lecture is just activation of T cells. So what will happen? The first thing, it will activate macrophages, and second thing, it will transmigrate. It will invite the WBC from the nearby blood vessel. Please come here. This is the infected tissue where you have to act. So all the WBCs, like including monocytes and neutrophils, they will come, and you know that monocytes basically get mature into macrophages. Monocyte, when they are present in the blood, they are not mature, and when they reach into the tissue, they get mature, and we call them as a macrophage. So these, with the help of these cytokines, these monocytes will start converting into macrophage, and furthermore these cytokine will attract the nearby WBCs as well. Okay, this is a type 1 Th1 first function. Now the second function is, it will activate this CD8 or cytotoxic T cell. So here is the CD8 plus cell. CD8 plus cell. It will be having TCR, and it is having MHC-1, sorry, CD8 protein. Okay, this interleukin 2 will go and bind over its receptor, and this will activate the T lymphocyte. Okay, so it means Th1 is basically activating the T lymphocyte. Now what will happen? This T lymphocyte will bind with this APC, but there is a problem. This CD8 protein can make bond with the MHC-1, but here you can see APC is having MHC-2. Now how this attachment will take place? No need to worry about. Let me tell you one more thing. MHC-2, this is the protein that is present on the APC. So this is the APC, and it is having MHC-2. B cell is the APC, so B cell will be having MHC-2. Dendritic cells are the APC, dendritic cell will be having MHC-2. But this MHC-1, it is present on all the nucleated cell of the body. All the nucleated cells of the body. So it means this APC, it's a basically granulocyte. It is also a nucleated cell. So this APC will be having MHC-2 as well as MHC-1. So it means on its MHC-1, it will again present the antigen. It will again present the same antigen that it has presented on MHC-2 to the Th0. It will present on MHC-1 to the CD8 or cytotoxic T cell. Okay, so this antigen will be recognized by cytotoxic T cell, and this is the stage 1 interaction. And now this stage 1 interaction will be further strengthened with the help of CD8. This is MHC-1, and we know that MHC-1 can make a bond with the CD8. MHC-1 can get attached with the CD8. So this attachment will take place. So now this is the first stage interaction. After the first stage interaction, we know that there is another interaction, that is with the help of B7, and that is B7 and CD28. So now this CD8 plus cell is now start proliferating. It will start dividing. It will start dividing. Now this, all the cells, all the new cells that are being formed from this side, CD8, they will be having ability. They will be having ability to recognize only this antigen. Only this antigen that was presented to this cell. So all these cells, they will be very specific. They will be very specific. It means that they will go into the infected tissue and they will search on this antigen only. They will be having their activity only against this antigen against which they has been activated. They will not work like this, that they will go into the tissue and they will start attacking on any antigen they will come across. No, they will be very specific in their action. So this is the reason these cells belong to innate or acquired immunity. Yes, acquired immunity. Because in the previous lectures I have explained that acquired immunity is very specific. It will act on it will act against only those antigens against which they has been activated. While innate immunity is non-specific. Innate immunity like macrophages, neutrophils, xenophils, natural killer cells, they are non-specific. They will start killing all the foreign particles that are coming into our body. Okay, so now CD8 cells has been activated. Now let's see how these CD8 cell will act. So for example, this is a cell. Let me tell you one thing. CD8 cells are getting activated by Th1, and Th1 was formed when this APC told this Th0 that the parasite, the pathogen is intracellular. When the pathogen will be intracellular, it will release interleukin 12. It will tell Th0 to get converted into Th1, and Th1 will activate CD8 cytotoxic T cell. It means this cytotoxic T cell is basically specific for the intracellular pathogens. Okay, so now this is a normal healthy cell. Okay, and it has been infected with the pathogen. Now, okay, it has been infected with the bacteria. Now what will happen, you know that every healthy cell has capability to digest or dissolve the invading foreign particle. Okay, so this cell will digest and dissolve this foreign particle, and it will present this antigen on its protein called as MHC-2 or MHC-1. MHC-1, because MHC-2 is present on the APCs, while MHC-1, this is the protein that is present on all the nucleated cells. Obviously this cell is a nucleated cell, so it will present this fragment of antigen pathogen on the MHC-1. Now this cytotoxic T cell, activated cytotoxic T cell, okay, this is the same antigen against which this cytotoxic T cell has been activated. So it means these cytotoxic T cell, newly formed cytotoxic T cell, they have ability to recognize this antigen. So now this cytotoxic T cell will come, CD8 plus cell will come, okay, it will make attachment with this antigen and MHC-1. Furthermore, you know that CD8 protein present on the cytotoxic T cell can make bond with the MHC-1. So this CD8 will make bond with MHC-1, and now this cytotoxic T cell will start releasing some enzyme. After this attachment, it will release perforin, and it will release Garanzyme B. Perforin and Garanzyme B. Perforin is a small protein that will cause spores in the cell. that will cause pore in the cell, and this enzyme B, it will penetrate into the cell. It will penetrate into the cell with the help of these pores, and it will start apoptosis process. It will start activating capsases. Capsases are basically proteins that are involved in the process of apoptosis. What is apoptosis and what are type of apoptosis, what are pathways of apoptosis, all these things will be explained in the later videos. Okay, so now it will cause activation of capsases and apoptosis of this cell. Now this cell will be died. Okay, so in this way the cytotoxic T cell works. Okay, now when these APC will tell this Th0 that the pathogen I have phagocytosed is basically extracellular pathogen, so you need to activate humoral immunity. So with the help of cytokine, it will tell this Th0 that the pathogen is extracellular, so it will activate Th2. Now this Th2 will activate B lymphocyte, and B lymphocyte will start making antibodies, and these antibodies will play their role in the humoral immunity. Okay, I have explained earlier as well that detailed mechanism of B cells and cytotoxic T cell will be explained in the later videos. What antibody do, how they neutralize, how they opsonize, all these things will be discussed in the next lecture. Today's lecture was basically related to the activation of T cells in the lymph node. So I am very much sure. Oh, let me tell you one more thing, that once this interaction has been formed, what do you think? The Th0 will keep on proliferating. Obviously this interaction has to stop after some time, otherwise there will be a hyperproliferation. We can suffer from a kind of autoimmune disease if there will be lots of T lymphocyte in the body. So nature has designed these cells very beautifully. So what will happen when this interaction is take place? Right after this core receptor interaction, there will be a signaling pathway, signaling mechanism to the nucleus, and a protein, and other protein. This was CD28. So there will be a formation of another protein, and the name of that protein is CTLA4. Name of this protein is CTLA4 protein. So what will happen? This CTLA4 protein has basically more affinity for this B7 protein, So after some time, when it will the signal will go, it will activate the CTLA4 gene. Translation will occur, and CTLA4 protein will form, and it will translocate on the outer surface of the cell. This CTLA4 has more tendency for B7, so what will it do? It will displace this CD20, the stage 2 reaction, and it will get attached with the B7 itself. When there will be attachment of CTLA4 and B7, there will be an inhibitory signal. And T cell proliferation will stop now. So what will happen? The same goes for cytotoxic T cell as well. So it has been seen in the mice. There was a experiment was conducted, and the gene for CTLA4 protein was removed. So it means those mice were unable to produce the CTLA4, and it was seen in those mice that there was hyperproliferation of T lymphocyte. T lymphocyte keep on proliferating because there was no inhibitory signal by CTLA4. CTLA4 was removed from those mice. Okay, so this is basically inhibitory protein, and it gives inhibitory signal for the proliferation. It was all about the activation of T lymphocyte in the lymph node. I am very much sure that you must like the lecture. Please don't forget to subscribe my channel and press the bell icon so that you may get notification for the future videos, and please don't forget to like and share my videos. Thank you so much.